Okay, so thanks for the introduction. I'm actually a PhD student in the University of Aberdeen. I'm going into my final year of my PhD, and today I'm going to be presenting some of my kind of preliminary results, looking into how palaeoecology can be used to improve our understanding of past changes in ancient woodlands in Scotland. So I'm sure kind of most of you already know that ancient woodlands in Scotland are defined as woodlands that have persisted since the first available woodland record. This is considered to be kind of 1750 CE or AD in Scotland. These sites generally have really high conservation value and associated biodiversity, and are considered to be our kind of closest example of a natural woodland in Scotland. Because of this, they're often used as kind of ecological reference points for woodland restoration and management. So when we're creating new woodlands, these kind of sites are often used as reference points, as targets that we can aim for. However, as you can see on the slide, many of these sites have kind of undergone a range of threats, from fragmentation due to changing land use, overgrazing, often with deer or sheep, lack of regeneration, often also linked to this overgrazing, and also excessive shading, and invasive species such as Rhododendron ponticum, pictured here. This means that these kind of sites could be giving us misleading perceptions of what is a natural woodland ecosystem. So we could be basing our restoration efforts on inappropriate references. So there are a few ways by which we can improve our understanding of long-term woodland changes. So firstly, we have our historical records, so things like our maps, texts, and photographs of sites. However, these kind of records are often kind of spatially and temporally fragmented. So areas that's of interest might not have very detailed records, and might be gaps in times where significant events just simply aren't recorded. Secondly, we have our kind of repeat long-term surveys. These can provide us with really in-depth details about vegetation changes in woodlands. However, in Scotland, or actually in the whole UK, our longest surveys are limited to the past kind of 100 years. So this means they're only actually looking at a single generation of long-lived trees species such as oak. Finally, we have palaeoecological techniques. This is things like the pollen, spores, and plant macrofossils that have been preserved in peat deposits. And hopefully I'm going to demonstrate during this presentation how these palaeoecological techniques are a really useful approach to understanding long-term woodland changes. The aims of my research are to provide stand-scale records of ancient woodland changes over time and look at the drivers of these changes. I'm also wanting to establish a chronology for these woodland records, so kind of a timeline of changes within the woodlands. This will help me determine whether the woodlands are truly ancient. And finally, I also want to look at the subcanopy layers within these woodlands, so the kind of shrub and the ground floor within sites. Typically, palaeoecologists kind of focus on the dynamics of canopy forming trees. However, long-term woodland monitoring has found that a focus on the canopy, which often appears to be stable, can mask substantial subcanopy changes. So how I'm going to do this, so I'm using a combined approach of using pollen and plant macrofossil analysis. Often these two techniques are kept really separate, but actually they really complement each other. So pollen analysis alone can provide us records on how vegetation around a site has changed. However, the pollen is kind of limited in taxonomic resolution. So not all species are identifiable, not all grains are identifiable to species level. For example, we have this cypressy pollen here. However, we don't know what kind of genus or species this pollen grain actually represents. So this is where plant macrofossils are really useful. The plant macrofossils are things like seeds, twigs, and buds, which are dropped from the plants directly into the peat. And these are usually identifiable to species level. For example, we have this cypressy pollen, and we can link this to specific Carex species. So it's just giving us more information about vegetation change within the woodland. Next, I'm going to use records from wet forest hollows. So these are essentially small wet depressions in the forest floor, which have been infilled with peat. And this peat then preserves the plant remains and the pollen. So typically palaeoecologists collect their records from lakes and peatlands, and these accumulate pollen from kind of tens to hundreds of kilometers away from the site. As the pollen is released high up in the woodland canopy, it's lifted into air currents and kind of transported across the region. This means these kind of records are amalgamating a broad range of habitats into a single pollen record. So for your ecologists and land managers, this isn't particularly useful when you're interested in a specific site, because you're getting a record of vegetation change across a whole region. In contrast, forest hollows kind of provide a really localized or stand scale record of woodland change. So within the woodland, the pollen, which is dispersed from the woodland canopy, is intercepted by trees as it travels through the air. This means that the pollen is transported much shorter distances. It's been found that kind of a roughly 1 hectare forest hollow can be dominated by pollen from a roughly 100-meter area around the site. Because of this, there's less tree pollens are deposited in the site, and the ground flora is then better represented within the pollen sequence. The peat also preserves the plant macrofossils, charcoals, an indicator of burning, and spores such as dung fungal spores, which are an indicator of local herbivory within an area. So these kind of using forest hollows is therefore a really ideal way for looking at woodland changes and the drivers of these changes. And my kind of a combined pollen and plant macrofossil analysis approach using these wet forest hollows is the first time it's actually been used in Scotland. So it could be really exciting. So I'm working in 4 sites across Scotland. These kind of form a broad transect. I've got 2 sites on the west coast, which are Atlantic oakwood sites. And today I'm going to be telling you about my results from Cambusurich Wood in Stirlingshire and central Scotland, and Dinnet Oakwood in Aberdeenshire in the northeast. So Cambusurich Wood is considered to be an ancient woodland in Scotland's inventory data. However, there's uncertainty of the origins of Dinnet Oakwood. The first record of this woodland is in the 1960s, and there's some suggestion that the wood might be partially or wholly planted. So there's a nearby place name which suggests there might be a much older history of oak in the area. So Fashnadaric is derived from Gaelic, meaning place of the oak. Generally, Gaelic is thought to have kind of gone out of fashion in this area of Aberdeenshire by the 15th century. So it'll be really interesting to see if there's a much older history of oak within this area. So the kind of bits highlighted in bold here are the results of radiocarbon dating. This is just telling us the age of material from the very bottom of each of the peat sequences I've collected at these sites. So the base of Cambusurich wood is placed in the Iron Age, and the base of Dinnet Oakwood is into the kind of late Bronze Age. So to kind of set the scene, this is a time period when woodlands had already substantially declined in extent in Scotland, which is partially linked to climate changes and also due to agricultural developments. So first, I'm going to show you some results from Dinnet Oakwood, and these are preliminary results, so it'd be nice. So here is my kind of coring site. This is the forest hollow. And this is the coring in process. So those of you who don't know how we collect these records, we essentially use a large kind of drill, which is then manually drilled down to the very base of the soil level. So until we get to like the rock and mineral layers. This here is the cores that are then extracted. These are then cut up, and the pollen and the plant macrofossils are extracted and identified in the lab. Once you've done all of this, we get these lovely looking pollen diagrams. This is actually a stripped back version of my pollen diagrams. It can be quite overwhelming, but hopefully I'll try and guide you through it. This is just showing some key taxa within this pollen diagram. So down the y-axis, we have the depth going down the core, which equates to time. And on the far left, we have the results of radiocarbon dating. As I mentioned before, the base of this record is in the Bronze Age. All of these silhouettes are then the proportion of each taxa within each sample depth. And here you can see there's little crosses as well, which is an individual pollen grain or spore, which indicates the rare types of the rare species within the pollen assemblage. All of these taxa have been split into different colored groups. So we have our tree species, our kind of sub-canopy trees, our heathland species, our herbs are in orange, and then we've got some ferns and sphagnum spores. And the ones here in brown are the dung fungal spores, which are indicators of local herbivory. We've got charcoal, a sign of burning, and this is just a summary diagram, so it's combining all of these little groups. On the far side here, we have the results of loss and ignition. So this is basically just telling us how much organic matter is in each sample. So a high value means there's lots of organic material within the peat, and when it declines, it means there's minerals in washing into the samples. Oh, and finally, All these diagrams are split into zones, which is just grouping together similar samples in different depths, just to hopefully help interpret the diagram. So first, if we look at the summary diagram, the combined pollens for trees and shrubs stays above 50% of the total pollen count for each of these samples. This suggests that Dinnet oakwood has actually been wooded throughout this record, so until the Bronze Age. And potentially Dinnet oakwood is actually an ancient woodland site. However, I'm going to demonstrate that the composition of the structure of the woodland does change over time. So below roughly 43 centimeters, we have much more closed woodland conditions with the greatest proportions of trees and shrubs. This closed woodland condition can be linked to declines in burning and local herbivory indicated by dung fungal spores at the base of the record. There's also potential signs of woodland management already quite far back in time in the record. So we have high levels of hazel or corylus and then followed by high levels of oak. This suggests these potential valuable timber species are being coppiced within the site. So we're already manipulating the canopy composition of this woodland by the late Bronze Age to early Iron Age. Moving further up the profile with the development of more open woodland conditions with the emergence of heathland taxa, so your Ericaceae, Micalluna vulgaris, and also increased herbs. These changes, so the woodland becoming more open, can be linked to more intense woodland pressures. We have increased evidence of burning indicated by the charcoal record, herbivores present, and grassland indicators. So we have our combined grass pollens and also Plantago lanceolata, which is considered to be an indicator of livestock grazing. And finally, at the top of this record, we have a return to much more closed woodland conditions. And there's also potential signs of planting. We have much higher levels of oak and pine, and also non-native fir trees coming into the record. This suggests a much more artificial tree species composition on the site. So this is the result of the plant macrofossil analysis from Dinnet oakwood. So this diagram is in a similar format to the pollen diagram, but each bar in this case is the number of taxa within each sample. And they also have the type of macrofossil at the top here. So whether they're seeds, bud scales, fragments of woods, or bracts. This record kind of supports the findings that we found in the pollen record. So the trees are present throughout the record. You can see them all going up here. And it's with a mix of kind of alder, birch, and oak. And these are kind of examples of the kind of things we're identifying here. We've got different birch bracts, and also this is how you identify tiny fragments of woods and twigs, which is in the record. This record is also providing us more information on the ground flora, which is directly around the coring site. In particular, is evidence of juncus species, which is not recorded at all in the pollen record. It's kind of highlighting one of the benefits of using plant macrofossil analysis as well. Juncus pollen is really susceptible to decay, so it doesn't really preserve at all in these pollen records. So this is the only indication that juncus is actually on the site. What's also really interesting is the charcoal record. So if you think back to the pollen record that we looked at earlier, they had peaks in burning at the very bottom of the record, and also in zone 4 here, where we had much more open woodland conditions. From the macrofossil record, which is larger chunks of charcoal, this suggests that this burning phase, the only main burning phase is at the very bottom of the record. And instead, in zone 4, the kind of increase that we saw in microcharcoal is just an artifact of the woodland opening up. So the smaller microcharcoal particles are being blown into the site, rather than this being a sign of more intense burning. And this kind of supports the idea that Dinnet oakwood was purposely conserved within a landscape of increasing human pressures. Generally, it seems that burning wasn't actually that important a factor in creating disturbance in Dinnet oakwood. Instead, grazing seemed to be more substantial. We saw evidence of grazing kind of throughout the record. Next, we're going to Cambusurich wood in Stirlingshire. Again, we have some photos of the site. This is the coring site here, the Forest Hollow. And this is the peat sample which I extracted. And this is just a nice example of one of the trees on the site. So it's a more mixed woodland. There's, as well as having oak, And there's also alder and birch and stuff kind of all mixed together within this canopy. Looking back at the pollen diagrams, this summary diagram, sorry, there's two main phases of more closed woodland conditions, right at the bottom and at the top of the woodland, at the top of this record. And this is when our combined trees and shrubs are above 50%. Whereas in the middle, we have the much more open woodland conditions. During this middle phase, there are substantial declines in trees, suggesting breaks in woodland continuity. This is especially clear in zone 2 here, where the woodland seems to be nearly completely replaced by grassland. Many of these events can be linked to different disturbances. So in zone 2, the changes seem to coincide with increased herbivore presence, indicated by these dung fungal spores. And in zone 3B, I think they called it, this peak in disturbance again is linked to burning activities, and also there's a rise, it corresponds to the rise in Rumex acetoizella, which is considered to be an indicator of livestock grazing. However, these kind of disturbance phases are short-lived. As we go towards the surface, the canopy structure within this woodland seems to change again. Generally, there's substantial decline in herbs and also the woodland shrubs, and it seems like the woodland is becoming much more simple in structure. This can be kind of linked to long-term grazing pressures. We see these long-term dung fungal spores in reasonably high, and also continued burning. This may also be an artifact of canopy closure, which is reducing light availability on the forest floor. So we're losing kind of these woodland herbs. Is this kind of more simplified structure which remains on the site today? There's very few woodland herbs within the woodland. If we look at the plant macrofossils, this is quite similar to the pollen record. We have increased evidence of trees towards the surface, and the herbs seem to be declining. Generally, it's important to note that most of the plant macrofossils are of wetland species. These are species that are generally within the forest hollow, which has been cored, rather than the surrounding kind of dryland taxa from the surrounding woodland. Woodland, we found, sorry. So what was I saying? So we found the legacies of past changes and disturbances has impacted the woodlands that we saw today. So the kind of stable woodland canopy that we saw in Cambusurich has massive substantial sub-canopy changes. I've also hopefully demonstrated how paleo techniques are really useful for testing if woodlands are truly ancient. This is particularly true for Dinnet Oakwood, where we find there is a long history of woodland on the site. However, it's not necessarily an oak woodland. Thank you for listening. If you've got any questions, I'll do my best to answer them.
Thanks very much. That was fascinating. Interesting analysis. Africa, a pollen core through a single grain of walnut, which just proves your evidence that this stuff can blow in from a long way away. So it is really fascinating. What I was going to ask, what occurred to me there was, do you actually compare this record with evidence of changes in the human population that would have perhaps influenced some of these changes that you've been talking
not
about?
yet, actually. So at the moment, I'm still waiting for the results of some of my radiocarbon dating. which will give me lots more information on the timeline of these changes. Then it'll be easier to link to kind of known historical events. I'm also hoping to then link it to known climatic events. So we have like things like the Little Ice Age and things, which is thought to have impacted woodlands in Scotland. And I'll see if I can be able to find those events within my records.
Yeah, obviously a lot more to learn.
Yeah.
Okay.
I'm still learning.
Yeah. Any other questions for Annabel?
Can I ask two really like very basic questions, which maybe other people will know, but your CE and BCE in the time, the dates, what do they stand for?
They're just, it's the same as saying AD or BC, I think it is. Yeah, it's just the same way of saying that. It just has slightly less like religious connotation. So academia, they like that kind of thing.
So it's a straight swap.
Yes, it's the same.
Okay.
Yeah.
And then the other thing was like, obviously you'll get this huge tube of peat. And then like, how do you get all the teeny wee fragments out? Do you just take a tiny bit, or like, is it by hand, or is there clever machines? Or I'm just interested to give a little flavor of the process of how you go about finding your betula seeds or whatever, and then having to decide which species it is, or, you know, I'm just fascinated by how that bit works, I guess.
Yeah, it's a very slow process. So for each of my sites, I've collected, both the cores are just about 1 meter long, so that's broken into 3 separate cores. And then for pollen analysis, cut it into 2 centimeter intervals. And then it's like a 2-day chemical process to extract all the pollen grains, which is just digesting all kind of the other materials which are surrounding the pollen grains, because it's really resistant material they're made out of. And then you have to identify all that under a microscope. And then for the plant macrofossils, it's kind of a similar process. You're cutting up all the cores again, and then you're doing a series of sieving. So you're just putting them all through a fine sieve, and then you pick out all of the seeds and then identify them all under a different microscope. And then for wood fragments, do certain cuts of the wood. So I use things like a razor blade and do really fine slices. So I don't know if you saw on the slide, there was one of the cell structures of wood. So it's just a lot of microscope work and a lot of cutting things up.
Thank you. Thank you.
Maz, you've got your hand up.
Oh, okay. I thought Coralie was first, but yeah, thanks.
Oh, I beg your pardon, Coralie. If you
I'll
were.
go just now. That was amazing. Thank you very much for that. But I was curious about bracken. Do you get to see bracken in your records at all?
Yeah, so bracken, it produces spores. So it's often kind of an indicator of when there's a denser woodland, there's often lots of bracken going on, or if there's lots of grazing and things. I don't think I actually included it on that diagram. There's so many different taxa, you have to cut it down to fit it onto the slide. But yeah, you do get bracken spores.
Thank you.
Coralie.
Hi, Annabel. That was really interesting, and well, I'm really pleased to see this sort of work going on that is looking in more detail than many pollen diagrams do at the sort of last couple of thousand years, because so many of the pollen records we have for Scotland are very long records, and they sort of only have a few sample points in the last couple of thousand years. And I'm a dendrochronologist, and I do woodland history work as well. So the sort of form and fate of our woodlands in the historic period is something where I felt that pollen could contribute more. And so this is more of a comment than a question, really. Sorry, Annabel, but I just wanted to say, I think this is really welcome, you know, both in the holistic approach that you're taking, but also that you'll be providing a lot more information about the last couple of thousand years for a number of sites. So that's really valuable. Thank you.
That was kind of like my aim. So I'm actually more of a ecologist in my background, but yeah, a lot of like the paleo work is just over such long time scales. It's not particularly useful for land management. It's not really useful to be applied, which is why I try to use the forest hollows to reduce the spatial scale, and also focusing on the last couple of thousand years, try and keep it as relevant as possible.
Absolutely. Well, I should look forward to your papers coming out, Annabel. Thank you.
In progress.
Thanks, Carly. Rose got a hand up.
Yeah, I was just going to ask, it looked on your map as though you've done another couple of sites somewhere out west, like along Loch Sunat. Could you say anything about those, please?
Yeah, it is very good map reading skills there. It's on Loch Sunat. They've got 2 sites on Arden American, just outside Salem and Arundel Oakwood.
Yeah, I used to work there, that's why.
Yeah. Yeah, the beautiful sites. Because of COVID, I've had quite a lot of delays, so those samples are still sat waiting for me. Hopefully in the next month or so, I'll be able to get working on them. And then I'm going to produce some papers out of that, and then a combined approach, which will be looking at climate in Scotland, because I have the kind of contrast of sites on the drier east coast compared to the west coast.
That'll be really interesting. I'll look out for those papers. Thank you.
Thanks, Ro. Alan McDonnell.
Hi, Annabel, that was really interesting. I just wonder, I know you're still getting through your PhD, but just wonder, are you drawing any early lessons for future management of woodlands?
Oh, good question. I guess one of the key things is the woodlands, the ancient woodlands that we see today are actually very different to how they were in the past. So Dinnet Oakwood, for one example, is completely dominated by oak, basically. And that's probably an artifact of more recent enhancement planting to get kind of valuable timber species. Whereas my record proved that in the past, it was a much more diverse woodland. There was actually a lot of birch and alder within the site. And also within Cambusurich, what's really interesting is in my record, I found lots of evidence of declines in the ground flora, so not many woodland flowers. And they've actually just started in Cambusurich to fence off areas to try and encourage regeneration and this restoration of woodland flowers. So kind of proving that what I'm finding is actually helping what's going on the ground, which is really great.
Thank you. Ro, your hand is still up. Is it an old hand, or is it, do you want to come
sorry,
back?
I've forgotten to take it down, that's all.
All right. Okay. Okay, super. Right. Annabel, doesn't seem to be any other hands left up. So I hope you enjoyed that as much as we all did. That was fascinating. Thank you very much for coming along and doing that presentation for us. And yeah, best of luck with it all and with your PhD.
Thank you. Thanks for all the great questions. Thanks.
You're welcome.